I think I've mentioned it a few times now. If you see something 3, 4, 5 times, then it's probably something you ought to get in your head. The idea was repetition, but hopefully hammer it in such a short time. So, T-cell receptor, one more time. It's a slightly different version of it, but you still have the alpha-beta T-cell receptor. It is associated with the CD3 complex, which provides the signaling that's required, and that's ITAS, immunoreceptor tyrosine activation mode T. So, tyrosine residues that become phosphorylated. I'm going to show you, and you've got it in your slides, T-cell signaling. And we're not going to get into it. I just want you to be aware of what's going on and why these ITAMs are important a little bit later in this lecture. All of this is acquired in the thymus, and then we go from there. This is just showing you another version of the, in this case, the class 1 MHC with the peptide in it and the T-cell receptor binding to it. You don't see the CD3 just for ease. But what we're doing here is we're looking down on the top. And what I want you to be aware of, and this is, if you really have to look hard to see what's going on here, but the kind of the opaque stuff on the bottom is the MHC molecule, and in there is a yellow peptide, not the 3. I probably won't be able to reach this, but so starts right here and goes down this way and up that way and up that way and up that way. Not these really bright yellow lines. That is a peptide in the groove, and it's from real experimental crystallography. And the T-cell receptor that binds to it is shown on top of it. And what they're talking about here is, remember the CDRs that we talked about, the 3 CDRs from the light chain and heavy chain. We also have them in the alpha chain and the beta chain of the T-cell receptor. And that's what it's showing you here. 1 alpha, 2 alpha, 3 alpha, 3 beta. That's kind of showing you where those molecules, those parts of the antigen binding site are recognizing. So the 3, CDR3 of both the alpha chain and the beta chain are mostly recognizing the peptide, but also part of the MHC molecule. I show you this because it really gets you to see that the T-cell receptor recognizes both the MHC and the peptide. If you don't have one or the other, the T-cell receptor doesn't get stimulated at all. Now, as far as MHC restriction, the first one here shows you, and this is just an example, but it gives you numbers so that you can follow it. So the peptide is X. The peptide is from the X antigen, the dreaded X antigen. And it is in context of a class 1 molecule, HLA-A201. This particular T-cell receptor recognizes the X antigen peptide in the context of HLA-A201. That's its restriction. If you take that same peptide, peptide X, and you put it in HLA-B5201, that T-cell receptor, even though it recognizes the X peptide in another MHC molecule, it doesn't recognize this complex at all. That's how tight the restriction is. It has to recognize both the MHC molecule and the peptide. We take the same class 1 molecule, HLA-201, in the final panel, and we put a different peptide in there. The T-cell receptor cannot recognize that either. It is that specific. That peptide with that MHC molecule. Even though that MHC molecule can bind 10,000 different kinds of peptides. So, very tight restriction. You've seen this before. The T-cells go to the thymus. They acquire what they're going to acquire there. They become an alpha-beta T-cell receptor positive and restricted to either class 1 or class 2. And then they go to the lymph nodes in the spleen. And we start talking about what's going on. Here's what happens. And this is where it gets fun. So, we have, step on something, that shows you a nail. So this person stepped on a nail. That introduced a pathogen into the foot. That pathogen has antigens. And you will have some macrophages that are in the foot, in the tissue there, that will immediately start to attack it. You will also have dendritic cells that are there that are going to pick up the antigens. So in order for the T cell to become activated following leaving the thymus, the first thing, one of the first things that has to happen is the antigen has to get to the lymph node for it to find it. And so I kind of mentioned this before. In the first 24 hours, it's kind of in this first little bullet here. The first 24 hours, the antigen can flow through the lymph to the draining lymph node by itself and get picked up by dendritic cells that are resident in the lymph node. But if you think about that, that's less and less antigen as the farther it goes. And so really the number of MHC molecules that are going to have that peptide antigen up on its surface is going to be pretty limited in the lymph node because the dendritic cells aren't able to collect very much of it. Those dendritic cells that are at the site of infection are surrounded by the antigen. So that it's very rich. They're picking up all kinds of it. And they're jumping into the lymphatic system and they're moving to the lymph node. But it takes them a while to get there. So dendritic cells at the site of infection are going to pick up this and they're going to take the antigen and process it, present it in context of MHC once they get to the lymph node, and wait for the T cell to come along to recognize it. We've mentioned this before. The naive T cell has left the thymus and is now moving around through the secondary lymphoid tissues. And it has on its surface something like L-selectin, which specifically binds to a marker on the high endothelial venule of the lymph node. So the naive T cell, first place it goes into is these lymph nodes, or the spleen, some kind of secondary lymphoid tissue. It is designed, the types of molecules, adhesion molecules on its surface, are designed to recognize the secondary lymphoid tissues and get into them. So the naive T cell gets in there. And we're not going to worry about glycan-1, CD34, and MAH00660. We're just, all we care about is the naive T cell gets into the lymph node across this high endothelial venule. Okay. This is just taking it another step further. And again, I don't care about these chemokine names that are up here. This is something that I go into detail in my other classes, but I want you to appreciate what's going on here because it's not as simple as a T cell floating along and bumping into the thing that binds to the L-selectin. And so this shows you a circulating lymphocyte. It's got L-selectin on its surface, so therefore it's naive. It's a naive T cell, and it's in the lymph node, and all of a sudden this L-selectin contacts, in this case, glycan-1 or CD34, and kind of slows down. But then it also has other receptors on its surface. In this case, again, we don't care, but it's CCR7, which is a chemokine receptor for a particular kind of chemokine. And there's lots of these. Lots of chemokine receptors, and we don't care, okay? All we care is that there are certain ones that are specific to the lymph node, and the naive T cells have receptors for them. So this is another example of the immune system is designed to get the right cell to the appropriate tissue. Multiple things involved in getting naive lymph nodes, or naive T cells into the lymph node, and not the activated T cells. So don't worry about any of the other things here. Just appreciate that there's more to it, more to the story, but we're trying to get the naive T cells and B cells into the lymph nodes. Once they're in the lymph node, so here comes the dendritic cell. It's popped in there, and it has the antigen on it in the lymph node, and it's in the T cell rich area. Remember that the blue area where the high endothelial venule is going to be the exit place for the. And I hope you appreciate that there's not just this hole. It's not a hole right there. That's a vessel going through the lymph node, and the high endothelial venule are just specialized endothelial cells right there. That's the right place for the cells to exit so that they'll be in the T cell rich area. So the dendritic cells are there. The T cells come out. They start sampling. And so the dendritic cells have a lot of surface area. They are class 1, they are class 2. And I'm going to show you what dendritic cells look like before antigen and after antigen in just a minute. And you'll get a sense of, okay, the MHC molecules go up. They express them a lot more. So there's sampling going on. The T cells are sampling, the dendritic cells for the MHC and antigen that they are specific for. And it really is as simple as going up and touching and seeing if there's a tight interaction or not. And if they don't find their antigen after a while, they go around and sample several of them, they'll leave. They'll go to a different lymph node and check it out. It's just an immune surveillance. They're just moving around almost randomly looking for their antigen. If they bump into their antigen, then they get stimulated and they start to proliferate. And that's the last panel you see there is one or two of those T cells ran into the antigen that they're specific for, and they started to proliferate. And that's right there in the T cell maturity as far as naive T cell. So now they've become an activated T cell and they're proliferating. The initial interactions, this is really just kind of get you a sense that it's not as simple as just bumping into each other, but there are these additional molecules. I'm not going to ask you to name CD2 and LFA1 and ICAM3. Just appreciate that there are additional adhesion molecules that make the interactions very tight. So there's a T cell receptor interaction, there's the CD4 or CD8 associated, and there are these other adhesion molecules. The most important molecule is coming up next. Besides the T cell receptor, I've already mentioned co-stimulation. Remember when I mentioned co-stimulation? We're going to talk about that here in a minute. Because naive T cells do not, the interaction between T cell receptor and antigen and MHC is not enough. That's the first thing they need to. They need that co-stimulation. But we have this interaction, we have nice tight binding with these adhesion molecules, and now we can start looking at possibly additional interactions. All right. Let's see what's next. This is it. I thought we were getting there. This is co-stimulation. Naive T cells are going to require the two signals to be activated. And you can see the first one is on the left side there, and there's actually a number 1 with a red arrow going up. That's signal 1, and that is our T cell receptor interacting with MHC, CD4, co-stabilizing that interaction. That's the first signal. The second signal is called a co-stimulatory signal. And in this particular case, there is a molecule on the surface called CD28. I do hope you appreciate this molecule. So CD28 is on the surface of the T cell. B7, its ligand, is on the antigen presenting cell. But B7 is not always up on the surface of these antigen presenting cells, and I'll tell you why when we get to it. So this is the second signal. You have to have the first part there, the T cell receptor recognizing its antigen, and you have to have CD28 on the surface of the T cell recognizing the B7 co-stimulatory molecule on the antigen presenting cell. Both signals required to get this naive T cell, keep this straight, naive T cell activated. Once it becomes activated, it doesn't need the second signal. So T cells, these naive T cells, are expressing CD28 constitutively. It's up on their surface when they're cruising around, when they're heading into the lymph nodes, whatever they're doing. It's already up. Antigen-presenting cells, like dendritic cells, do not have B7 on their surface when they're just hanging out. They don't express them constitutively. They have to be induced. So B7 molecules are expressed and put up on the surface once they come in contact with a danger signal. That's the important thing I want you to understand, that something tickled those dendritic cells and made them think that there was a danger signal. And what those things are? Back to the very first thing we talked about in the first hour, PAMPs and PRRs. The pathogens have certain things like LPS, certain conserved motifs that are recognized by these receptors on the surface of our effector cells, the PRRs, that would stimulate the antigen-presenting cell to put B7 up on the surface. So let me get that, let's go through this one more time. The T cells already have CD28 on their surface, but they do not see B7. If they don't see B7, we'll see what the consequence is. Antigen-presenting cells don't have B7, the ligand for CD28, unless they have received some kind of danger signal at some point during this process. They are going to see that in the case that we saw a while ago where a person stepped on the nail, pathogen got introduced. There are some PAMPs there, whether it's LPS, teichoic acid, CpG DNA. Something is going to stimulate that dendritic cell to put the B7 molecule up on its surface so that it can co-stimulate the T cell recognition. Danger signals, here we are. Phagocytosis breakdown of bacteria by macrophage induces expression of class 2 and B7. So it puts up both of them. Some of those products, LPS, CpG DNA, etc. We talked about a lot of these earlier on, toll-like receptors recognizing various ones. Those are the ones that I always talk about, LPS, because all the gram-negative bacteria make lots of it, and it's very inflammatory. It just causes all kinds of inflammatory reactions and will certainly cause B7 to go up on the surface. So the danger signal induces in this case, a macrophage or a dendritic cell to put MHC on the surface and the B7 molecule up on the surface. Let's see what else.
So does the B7 CD28, that activates the naive T cell, but then the MHC molecule presenting the peptide shows it what it is and what it needs to attack?
It's the first. There's actually two signals going on there. The first signal is the T cell receptor. Let me see. The first signal would be the T cell receptor, and you remember it has CD3 with all those ITAMs and things. A lot of signaling motifs involved, much more than even the CD28 and B7 thing does. That is the first signal. Okay, that's a signal to the T cell. And this is a critical thing to get down because two signals are required for a naive T cell. And if we don't get the second signal, you're going to find out that another thing happens, and that's anergy. We basically will kill that cell without that second step. So the first step, and the bottom part of this shows a 1 pointing down. That's the MHC and antigen interacting with the T cell receptor. It's stabilized by, in this case, it's a CD4 molecule. That's signal 1, and it's a very strong signal through all of those ITAMs and CD3. And then there's a second signal that's not as strong but is important, is the B7 binding to CD28 and particularly the CD28. Now, you can envision mimicking interaction with B7. Say you've got an antibody to CD28 that will bind in such a way that it could cause that second signal. So you could bypass the need for a danger signal. But these things that we're talking about, these danger signals, remember when I talked about adjuvants? And I said that most of the adjuvants that we've used in the past for humans have been alum mixed in with the shots that they give us. They're getting a little more sophisticated now. In animal studies, we use nastier things. I knew a guy he was a vaccinologist back at Tulane Medical School. He swore that an adjuvant just had to irritate. It just had to be an irritant. And he says, I bet I can give an antigen with Coca-Cola up the nose and get an immune response. That acid going up in the nose was enough to inflame the antigen-presenting cells in such a way that you could get this co-stimulatory stimulation. So some kind of signal to the antigen-presenting cell that this is a bona fide antigen that we need to respond to. T cell receptor is the first signal, the co-stimulation is the second signal, and that will get the T cell fired up. So how does the dendritic cell get this? This is what a dendritic cell looks like before and after on the left. And you can see on the right, that's a transmission electron micrograph, and it shows all the pseudopodia, or whatever you want to call those things, sticking out. Lots of surface area. So immature dendritic cell has on its surface, and the ones that you need to care about are MHC class 1 and MHC class 2. Don't worry about CR4, those other things, adhesion molecules. And DEC 205, all that is it's something that helps it uptake. So the cell on the top, what you need to be aware of, the cell on the top is designed to take up antigen. It's designed to pull in antigens and process them to go up in the context of MHC. Once it leaves the peripheral tissue and gets this danger signal, these other things come up. And what you should notice, and it's not very evident, but more MHC class 1 and more MHC class 2 comes up. The B7-1 and B7-2 come up. Those are the two green things that you see right there. And some of the uptake things have gone down. So it's kind of switched from being an antigen uptake cell to being an antigen presenting cell. And it has the co-stimulatory molecules that you need. All of this happened because it was out there in the tissue. It came in contact with a pathogen with a PAMP that it recognized, and it became stimulated to become antigen processing and put these co-stimulatory markers up on the surface. Don't worry about the other things on there, but they play a role, not for our purposes. What happens if you don't have it? Part 1 on the left, we have our 2-signal situation, and the T cell gets activated. T cell receptor, MHC, it's stabilized with CD4 because this is a class 2 situation. That's signal 1, co-stimulation through B7 and CD28, and it causes signal 2 and we activate the T cell. If we don't get that second signal, the T cell becomes anergic. And I've told you energy or state of energy is unresponsiveness. It's not dead yet, but it will not respond. It can run into its antigen 100 times. It's not going to respond now because it didn't get that co-stimulation the first time, and it's an anergic T cell. This is important because if you do have self-reactive T cells, as an example, that get out of the thymus, and they bump into the antigen and the MHC molecule that they're specific for out in the periphery, you don't want it to respond. But if there's a co-stimulation going on there, it will respond and you'll have a nasty autoimmune disease get started. So self-tissues out there that there's no pathogen, there's no need for there to be any kind of co-stimulation, there isn't going to be, and those cells don't get reactive. This is the case, though, that we see in some, it's starting to show up some in autoimmune diseases. In that you have a virus infection that may cause a self-reactive cell, T cell, B cell, or both, to become stimulated inappropriately. And that's what drives the autoimmune reaction in the first place. When it should have been, you should have been tolerant, you should have been able to regulate that response. Because there's an infection going on, you get an inappropriate autoimmune reaction taking place. We're not there. We won't go there. Okay, what's next? I forget what this is. Oh, that's kind of looking at that a little bit. So in the top part we have a macrophage, and it's just picking up some protein. Nothing to worry about. Maybe it's a cell protein. Maybe it's just dirt. It's not dangerous. It doesn't stimulate the B7 molecule to go up on the surface, and so when it interacts with the T cell on the bottom panel, we don't get the second signal, and so that T cell becomes, it doesn't respond. In the second one, we have bacteria, and we have the appropriate thing taking place. B7 goes up on the surface, and we get the signal taking place, and we respond. The T cell that's specific for this bacterial antigen responds, and we have a good response. In the last column, you mix them together, and so you have a non-dangerous antigen that gets pulled into the same time with a dangerous antigen, come in contact with a T cell specific for the non-dangerous antigen, and because that antigen presenting cell did run into a danger signal, it does have the co-stimulatory activity. And so here you have a case where maybe a self-reactive T cell typically would not be stimulated like in the first panel, but if by accident that antigen was presented on an antigen presenting cell with co-stimulation, then you could get activation of that T cell to a self-antigen or to something that you don't want to respond to, something that's not necessary to respond to. So it's kind of a collateral damage type thing that can happen in cases. This is something I brought up that I think it's very important for you to understand, because it could play a very important role in the anamnestic response. So before you go through an immunization, the primary response is you get a booster and you go through a secondary response, etc. You don't have very many B cells specific for your favorite antigen. Once you've immunized with your favorite antigen, you've expanded the population of B cells specific for it. So there's more of them around. They're in the lymphoid tissues, hanging out. And if it happens to be a T cell dependent type antigen, like a protein, then you would have done the same thing with the T cells, and there'll be more of those. And so there are probably those memory cells hanging out in the lymph nodes or the other lymphoid tissue. But in that early part, the second sentence, third sentence, too few antigen-specific B cells to present antigen to naive T cells. So there's just too few of the B cells, too few of the naive T cells for them to ever come in contact with each other, in this case for the B cell to be the antigen-presenting cell, because B cells only present antigen they can take up through their own B cell receptor. Therefore, they only present antigens that they're specific for. Does that make sense? They don't have the mechanism to take up a gram-negative bacteria and process it and present it. They have the ability to bind to a toxin and internalize it and break it up and put it up on the surface. But they have to be specific for the toxin, or at least part of the toxin. They're specifically part of it. But as you go through this, the immune responses, more of those guys are present, more of the T cells are present, they interact much more, and this type of interaction as B cells, as presenting cells, can be much more important in secondary immune responses, and you would get going fast. You have a memory B cell and a memory T cell hanging out right there together. The antigen comes in, the B cell grabs it, hooks up with the T cell, and the T cell gives cytokines. You would have a great response going crazy right off the bat. This is why you get tetanus vaccines so many times. We are continually trying to keep our populations of T cells response specific for the tetanus toxin and the T cells that would help those responses, make sure that they're around. In fact, you also want there to be circulating antibodies. Now, you're not going to have a whole lot of them, but if you step on a nail and you've had DPT vaccines, or you've had lots of vaccines through your years, all you have to do is, okay, I stepped on a nail. Well, it takes a while for Clostridium tetani to get deep into, it's an anaerobic organism, it has to get deep into the tissue in an anaerobic state, and it'll start growing and then it'll start producing that toxin. You have enough time to go to your doctor or your PA and get a vaccination, another DPT vaccination. Boom, those B cells start going, those T cells start helping them out, and the antibody starts flowing before enough of that toxin starts getting produced. So that's why we do that. And that's one of them that we really are very critical about, because like I said, that toxin is just bad. It would, somebody said a thimble full of it put in Lake Thunderbird would kill everybody in a moment. It's just, it is so potent. It's one of the most potent toxins to man. There's another one that's a little worse. Its cousin is Clostridium botulinum, causes botulism. That's a very, very nasty toxin as well. And they will kill you in very, very low doses. So anyway, I digress. Okay, now we're going to talk a little bit about signaling, just because I think it's cool. I just think this is really cool, but also because Dr. Latassi wants to talk about kinases and inhibitory drugs and things like that, and some of that's going to be seen in here. So I don't expect you to know any about it. I just want you to appreciate what's going on here. And the little red dots that you see in the second panel, that's a phosphoryl group attached to a tyrosine residue. So in the first panel, those ITAMs, those immunoreceptor tyrosine activation motifs, are unphosphorylated. The T cell receptor binds to its antigen in MHC, stabilized by CD4 at some point, and those ITAMs get phosphorylated by ZAP7, by FIN or LIC or one of these others, these kinases that you see down here. So kinase, if you don't know what kinase is, it is an enzyme that adds phosphoryl groups to proteins. In this case, it's a tyrosine kinase. And so the signaling that takes place is those tyrosine residues get phosphorylated, and that's going to allow additional molecules like ZAP70 that you see, the little yellow thing way down at the bottom, is now going to be able to come up and dock on those. And then you see the LIC, which is another molecule, come over in closer proximity to the ZAP70. These domains open up and you get the LIC kinase will phosphorylate the ZAP70. And then as we go through this, the ZAP70 leaves and goes and does something really cool. Here's the other steps. LIC phosphorylates tyrosine residues on the CD3 and ITAMs. LIC activates ZAP70, blah, blah, blah. PLC gets done, blah, blah, blah. Diisoglycerol gets made. In the end, signal goes to the nucleus to turn on certain things. So, but all of those things are therapeutic. For your purposes, if you want to start thinking about in the future, how do we shut down T-cell responses, maybe a drug gets developed that shuts down the LCK or LIC kinase, and you just give that to them, and they're not able to make T-cell responses because the T-cell is no longer signaling to that. One of the molecules that Dr. Latassi is talking about is the JAK kinases. Are y'all familiar with the JAK kinases? The JAK kinases, there's 4 in the family, and I won't go into the specifics whether JAK 1, 2, and 3, and then there's something else. But they are associated with a lot of the cytokine receptors, and you guys have heard me talk about IL-1 beta, TNF alpha, IL-6. There's other pro-inflammatory cytokines like IL-10. I think, I don't know what else has been mentioned, but there's IL-12, and I'm going to go into some of these kind of cytokines in a minute, but there's GM-CSF, and GM-CSF is granulocyte macrophage colony stimulating factor. It is a cytokine that is made, it goes back to the bone marrow and tells the bone marrow to make more neutrophils, mostly. Some macrophages are most of them, but it's a granulocyte macrophage colony stimulating factor. Basically, we need more neutrophils to fight this infection. But the JAK kinases are important signaling mediators. Like you see up here in some of these things, we've got a LIC kinase, we've got some of these other things. The JAK kinases are associated with the cytokine receptors. So IL-1, TNF, IL-6, we can go on, IL-10, a bunch of these cytokines that are involved in inflammatory responses. The receptors that they bind to are associated with JAK kinase. So JAK kinases play a role in the signaling through those cytokines. And I just mentioned that because I know that that's going to come up later in what you guys are talking about. And I want you to be aware that JAK kinases play a role in signaling through some of these cytokine receptors that we've been talking about. All right. Once we're activated, so the T cell has interacted with a dendritic cell or a B cell and received a naive one has received both the T cell receptor signal and it has received the co-stimulatory signal. They have become activated and they leave the lymph node and go look for their particular antigen. They no longer require that co-stimulation through B7 and CD28. Once they're activated and they go out and search for their antigen, all they have to do is come in contact with their antigen and they have the ability to do what they do, and that is produce cytokines or be cytotoxic. And they don't need that CD28 to do that. But there are other molecules that could play a role and help the response work a little bit better. That last panel I'll just mention is showing you a CD8 T cell. And what you have there are host cells, and they're trying to show you that 2 of them are infected with something, most likely a virus. And what's going on there is that's a naive, or it's not a naive, that's an activated cytotoxic T cell, the CD8 T cell. And it is going out and it recognizes its specific antigen on one of the self cells that are out there, and it binds and delivers the cytotoxins that kill those things, so the viruses can't replicate. And this is just kind of going into that in a little more detail. Here you have a CD8 T cell. When it's naive, it has to interact with a dendritic cell that in this case is infected by the virus and has put a viral peptide up on its surface in class 1 through the endogenous pathway of antigen processing. It also has received the danger signal, so it has the co-stimulatory molecules up there. CD8 T cells actually require a little stronger stimulation than the CD4 T cells, and that's for good reason, because they are very destructive. They like to kill things. So, but in this case, we get the signals required for the naive T cell to become activated, and now it goes and it starts killing. And so in the bottom, you don't want this thing just going around killing things, so if it goes out and it runs into something that's specific for on a self cell, it doesn't just respond by itself. The naive CD8 T cell is not just going to dump its granules and kill everything that comes in contact unless it's a bona fide danger situation. We have a virus that we need to take care of. All right, other activation molecules, not a whole lot more to go, and then we'll open it up to whatever y'all want to do. There are other ones. I do think that you need to be aware of this, and the next slide actually shows a picture of this, but there are other membrane molecules besides the CD28 and B7 interaction, such as CD40 on macrophages binds to its ligand, which is called CD40 ligand on the T cell, and you get more production by the T cell. Now in this case, it's probably already it's activated T cell. The CD40 ligand interaction just really soups up that interaction, makes that T cell do its job even better. In the case of cytotoxic T cells, there's these molecules called FAS ligand, and that co-stimulation of that interaction helps the T cell cause apoptosis or death of the target cell. And I bring these up because these are areas that are actively being researched as targets for immunotherapy. Antibodies to CD40 to break that ligand. Or maybe you want the T cells to respond more. You have an agonist type antibody that binds to CD40 ligand in the T cell and tickles it, and basically mimics the reaction it would have with the macrophage, so it produces more cytokines. Or maybe you've got the cytotoxic T cells out of control, and the FAS ligand binding to FAS is making it worse. How do we, what can we do to block that? And antibodies or some other kind of therapy might be useful. And that is some of the stuff that's being researched right now. This is just looking at those same things in a diagram, and I like this one because it has some of the cytokines at the bottom that I think you ought to be familiar with. The left panel are the cytotoxic T cells. In this case, we have MHC and interacting with the T cell receptor, FAS ligand on the CTL binding to FAS on the virus-infected cell, and it causes the cytotoxic T cell to release things like interferon gamma, TNF alpha, don't worry about TNF beta, and then the one that I want you to be aware of on the other side, from the granules, is perforin. Perforin and granzymes, really. Perforin is much like the complement. I told you about complement, and in the end, it can form a pore. It can polymerize in the membrane of a pathogen and cause these pores to form. Perforin can do the same thing. It basically can make a hole in the cell, but in this case, the purpose is to get the granzymes in, and the granzymes go in and cause apoptosis in most cases. T helper 1 cell is going to interact with the macrophage. CD40 ligand binding to CD40 is going to help it to produce cytokines. And if it's a T helper 1 cell, which is I'm getting ready to go into here in a minute, it's going to produce gamma interferon for the most part, but it can produce some other cytokines. Like I think it's got what else is on there? Others, it's got IL-3, TNF IL-2. It's really going to be gamma interferon and some IL-2 that we think about with those types. The gamma interferon is a great macrophage inducer. It's the best at making macrophages angry and do their job a lot better. The next one, T helper 2 cells, and I don't know if I've really gotten into T helper types yet, but we're getting ready to do that. These are the two types of cells, CD8 and CD4, that we think about with alpha beta. Well, we can break the the CD4 cells into even more smaller subsets. T helper 2 cells help these cells make antibodies better, and they produce IL-4 and IL-5 and things like that to stimulate those types of responses. How do we regulate these things once they get going? So in the first slide, we're actually showing the co-stimulation that we've talked about before. T cell receptor binding to MHC, CD4 stabilizes, and the B7 on the antigen presenting cell is sending a signal to the CD28 on the T cell. You get the two signals together. Well, there's another molecule that gets upregulated on activated T cells after a while. After you have the T cell has been stimulated so long, CTLA-4 moves up on the surface. And so what you're seeing is the pink thing. Where's my little pointer? So you still have the CD28 here and here, but now you have CTLA-4. And CTLA-4 actually binds to the B7 molecule in a dimer that is much tighter affinity. It's at 20 times there. It binds at a 20 times higher affinity than does the CD28 binding to B7. And let me move on from that. So this shows you B7 on the left binding to CD28, the CTLA-4, which comes up on the activated T cell, dimerizes with the B7 molecules, and what it does is it sends an inhibitory signal. I don't know if I've even put that in there. The inhibitory signal from CTLA-4, get ready for this, you're going to love it, ITAMs. So we had ITAMs, immunoreceptor tyrosine activation motifs. What's the I going to be for here? Inhibitory. And so these are tyrosine residues that when they get phosphorylated, they will shut the T cell down. We're going to regulate the T cell and make it stop doing what it was doing. It's a great way to regulate. I've already told you this once. What's the best way to regulate an immune response? Get rid of the antigen. Somebody said it, I heard it. Get rid of the antigen. As long as the antigen's there, the immune response is going to continue to be mounted. Okay, real quick, what about gamma delta T cells? Antigen recognition here is most likely through some non-classical MHC molecules. There's some additional MHC molecules that we didn't talk about, different parts of the chromosome that may present phosphocarbohydrates or something like that on these non-classical MHC molecules to the gamma delta T cells. Not really sure, we don't really care. They may be more of a first-line defense, but it's more specific than the innate mechanisms do. And they may play a critical role early on, but we don't really worry too much about what they do. I did mention superantigens, I think. Superantigens such as toxic shock syndrome, toxin 1. You can see on the left panel, it's really showing you this TSST and also another type of antigen, I think from another streptococcus as well, an erythrotoxin. They're superantigens in that they don't bind in the groove and cause one particular T cell to get stimulated. They cause a greater number of T cells. They're non-specific because they bind at a different place in between MHC and T cell receptors that are not specific for them, and they cause a polyclonal type of stimulation. Another one would be some of the viral superantigens. Now, I don't have an example of those, but there are some out there called superantigens, and they cause polyclonal stimulation that you may have heard of. All right, let's do a summary of T cell activation. We started here at the top. we have, these two things are going on all the time. We're getting infected with something, the dendritic cell in the neighborhood is picking up the antigen is moving to the lymph node on its own, but a dendritic cell is picking up great gobs of it, processing it, putting up more MHC molecules, putting up co-stimulatory molecules if there's a danger signal, and moving to the draining lymph node. The activated the naive T cells have the L-selectin on their surface. They're rolling along until they hit the high endothelial venule, and they cross over, and they move into this T cell rich area and go in search of their particular specific antigen. In the case of the cytotoxic T cells, they're going to recognize the MHC class 1 on the dendritic cells. They're going to be stabilized by CD8 and will have co-stimulation that will cause them to become activated and then proliferate. Then they will leave and they will go in search of other self cells that are infected with the same viral particle and kill them. On the case on the right, we have the CD4 T cell recognizing the context of MHC class 2 co-stimulation through CD28 and B7, and we get proliferation. Now the activated T cell no longer binds to the high endothelial venule and goes out and looks for, it either leaves the lymph node and goes and looks for its specific antigen and bumps into a macrophage in the inflamed tissue where the infection took place, and you get activation of the macrophage to kill better. And the final one, the T cell may not leave the lymph node, it may come in contact with a B cell specific for its antigen, and that's if it's a T helper 2 cell. And that causes differentiation into plasma cells that then make antibody. What I want to do next is I want to just go up here on the board and I'll talk about some of these T helper types that you may or may not have heard about. So if we're talking about the CD8 T cells, we'll do this real quick. CD8 T cells recognize class 1. And what they're going to end up doing is they're going to be cytotoxic, and they do this through the release of porphyrins and granzymes, and they're going to kill these cells. They also will produce some interferon gamma. Now, the ones that we're going to focus on are all over here called CD4. And we're going to start with T helper 1s. We can break these down into T helper 1s, and you might see it as T little h1. It doesn't matter. It's T helper 1. We have a T helper 17. There's not 17 of these. It just makes IL-17, so they call it T helper 17. T helper 2, you've seen these two. We have one called TFH, it's a follicular, we don't care much about it. And then we get down here, T-reg, and I'll get to it last. So how do you end up getting a T helper 1 response? And this is something that we're going to talk about in more detail tomorrow, but when that dendritic cell is in the tissue and it's interacting with a pathogen, that pathogen has the PAMPs that are binding to certain PRRs. And I talked about this a little bit. Those innate recognition mechanisms are somewhat specific for a type, but not specific to the antigen level. So a certain toll-like receptor recognizes CpG DNA. That's found in bacteria. Certain ones recognize LPS. That's found in gram-negative bacteria. Those things aren't found in viruses. Those things aren't found in yeast. They're not found in the big parasites. Other PAMPs are found in those. And each one of these PAMPs will bind to a PRR, pattern recognition receptor, and stimulate that cell to produce certain cytokines. So you have an environment. You produce the environment. So if the dendritic cell bumps into a gram-negative bacteria, it's going to bump into LPS. And so that PAMP's going to be recognized by the pattern recognition receptor, and it's going to cause the dendritic cell to not only take up the antigen and start processing it, but it's also going to gear up the dendritic cell to start producing certain cytokines that PAMP tends to stimulate. And in this case, it could be something like, we'll just put some of these out here. What I'm going to put here is cytokine environment and in parentheses trigger. So what this is going to be is, if this T cell, this T cell is CD4 positive, it recognizes an antigen in the context of class 2, and it has CD4 on the surface that stabilizes that interaction. It doesn't know that what it recognizes is a bacteria or a virus or a parasite or whatever. But the virus, the parasite, the yeast, the bacteria have the PAMPs that stimulate certain cytokines to be released by those dendritic cells in the environment when it's interacting with the T helper cell. And so, depending on the environment it's in, it will stimulate the CD4 T cell to become one of these. Not the Treg, we'll get to it later, but one of those 4. So if there happens to be in the environment IL-12 and interferon gamma, that's a trigger for that CD4 T cell to become of what we call the Th1 phenotype. And that Th1 phenotype is, it happens to produce IL-12 and interferon gamma itself. And that, what's the function is activate macrophages. And this is that's the way you say macrophages in shorthand. So, where do these come from? This actually comes from macrophages, IL-12. So there's a macrophage handy in the environment, and it has come in contact with a PAMP that causes it to produce IL-12. This actually comes from an NK cell, is what we think. A lot of this is just conjecture. We're just guessing where they're coming from. But we happen to know that if we take a CD4 positive T cell that is not committed one type or the other, if we grow it in the presence of IL-12 and interferon gamma, it becomes a T helper 1 cell, and it produces IL-12 and interferon gamma. If we were to grow the same cell in the presence of IL-6 and IL-21, those types of cytokines, also IL-5, is that what it does? Yeah. No, I'm sorry. Yeah, that's right. No, here's that. Can't even read this. IL-6 and IL-21. Then this one, what it does is it produces IL-17. That's the predominant thing that it produces, and that's why it's called the T helper 17. IL-17 recruits and enhances neutrophil responses. And so it's going to be very pro-inflammatory, and it's causing the neutrophils to go and cause more damage. T helper 2 cells, if you grow these cells in the presence of IL-4, they will then start to produce, and the trigger will be IL-4 and IL-5. And what these do is they activate cellular and humoral or antibody anti-parasite responses. So a response to parasites. Now, what does that mean? Let me just kind of go over these. We're not going to worry about this one. This one it's a follicular cell that is involved in B cell responses. What this means is, in this case, the same type of cell in this environment is now going to differentiate into a cell that produces IL-12 and gamma interferon. And what it's going to do is it's going to go out and it's going to find macrophages out there and provide gamma interferon to them and make them much better at what they do. In this case, if that CD4 T cell is being exposed to the cytokine environment that includes IL-6 and IL-21, it's going to, one of the things it's going to produce is going to be IL-17. IL-17 can do several things. One of the things is it kind of can stimulate more production of neutrophils, but it also can enhance the neutrophil response. In this case, IL-4, we get a T helper 2 phenotype, which is IL-4 and IL-5. What those do is they cause an isotype switch to IgE. That's one of the things that IL-4 and IL-5 do, is cause the B cells to switch to making IgE antibodies. And those IgE antibodies then go and bind to mast cells. And so it's really here for expulsion of parasites. The mast cells hanging out in our tissues come in contact with they have these specific IgE antibodies bound via Fc receptors, and they become triggered and they release their granules, and it causes the responses that we have that would involve or become expulsion of the parasite, but most of us know it as allergic reactions. That's what we get out of it. We're not going to worry about the follicular cell, but the Treg cell has been so controversial for years. We used to call certain cells T suppressor cells, and a lab I worked in worked on them for many, many years, and these were fighting words. Scientists that were really into studying T cells would really get angry at each other because some said T suppressor cells didn't exist, some said they did. What we've come to find out is that there are certain kinds of CD4 T cells called Treg cells, and what they do, they really come about they really kind of come about in the thymus, but they can be kind of produced outside the thymus if you have enough TGF-beta around. TGF-beta is transforming growth factor beta, and it does some things, including, I won't talk about that right now, but what this one will produce is some more TGF-beta and also some IL-10, and just as its name thing says here, it regulates other T cells. The TGF-beta will certainly inhibit some of the responses that you might get from other T cells, but I wanted you to kind of start to visualize. We've talked about T cells a lot, we've talked about activating T cells a lot, but it's not just what MHC do you have and do you have co-stimulation or not, it's what environment are you in, and that environment is dictated by the type of pathogen that you're trying to respond to, or if it's not a pathogen, if it's a self-antigen or something along those lines. So there's that. I think that I'm going to stop right now and open the floor up to you guys to ask questions. Now that Dr. Cassie's here. I've already told them what we're doing tomorrow. They say they need to cogitate and other big words like that and think about this. And we'll do the anatomy of an immune response tomorrow, and then they will stick around in the afternoon and ask questions if they want, or go take a nap if they want, I guess. Because they don't have anything else in the afternoon?
You're it.
Whoa, no pressure. Any questions? Had it? Got it? Yeah.
Should we memorize this chart?
Memorize it? No. What I want you to be aware of is, well, I want you to, I use the word appreciate. I want you to appreciate that the cytokine environment that they're in affects what they become. And that's one of the things that we haven't talked about. We've just been talking about T helper cells, cytotoxic T cells. There are different types of T helper cells that do different things. And the cytokine environment that they are induced in or activated in affects what type they become. And I do want you to do know this. T helper 1 cells activate macrophages, T helper 17s, enhance neutrophil responses, T helper 2 cells. I would just say help B cells make antibodies, make better antibodies. Because they produce cytokines that are typically thought of as being B cell cytokines for better activation, better, well, for one thing, proliferation. And when you proliferate in a B cell, you have the potential to go through affinity maturation. So those are the cells that would be responding. Those in the TFHs are the ones that would be interacting with the B cells to cause them to proliferate and make better, more specific antibodies. Everything all those ways goes through. Yep.
I have a kind of a random question. If you were in our shoes, how would you best utilize, like, would you suggest reading through these and then going back through the PowerPoints, or how would you suggest, like, us breaking up our time to best digest and understand?
That's why I gave them to you, because I thought that the important things were in the handouts and in the slides, but there are also some things in the slides that are overkill.
Okay.
I tried to point those out to you.
Okay.
You don't need to know that chemokine receptor 7 is on the surface of epithelial cells in this particular case, and those kind of things are not important at this stage. Yep.
I had a question about when a cell becomes anergic, it stays like that. It has one chance to have the secondary trigger, and then it's done.
That's the way I understand it.
Okay.
Yeah. And then they will eventually just die
Okay.
because they will not be able to get the survival signals that they need, because these types of cells require some kind of stimulus over time. And in the case of T cells, interleukin-2 is the main cytokine for causing proliferation, for them to make more of themselves and to stay alive. And if they're not getting, if they're allergic, they're not going to be able to, they're probably brought their IL-2 receptors down and can't respond to them. I don't know, but allergic means unresponsive. They will end up dying. Dying of neglect, basically.
Okay.
Yes?
What's the difference between B7-1 and B7-2?
There is a difference, but we're not going to worry about it.
Okay, so keywords.
B7, just call it B7.
What about 41BB/41BB?
That's another co-stimulatory molecule that's another way to induce CTL, the cytotoxic T cells, and we're not going to worry about that one either. Yes. I don't even remember that being on the slides.
I don't think I've ever heard of that in my life.
And then talking about non-classical MHC molecules, I'm not really sure.
Okay.
I don't remember which ones you're supposed
They're,
to say all the things one more time.
Summary of today.
so the only MHCs we're going to be that we worry about are those 6.
Oh, okay.
The non-classical aren't even encoded at the same loci. There's another whole section of genes that are non-classical MHC genes. They're like MHC, but they don't do the same types of things, and we don't care about them. We really don't. They're so minor in what they do. These are significant in what they do. The non-classical are, in some cases, they're actually called minor histocompatibility complex antigens.
About 6 slides ago, where it has the CD8 T cells and the CD4 T cells and all their associated cytokine molecules, you said be familiar with those. Should we be familiar with IL-4s specifically to like the T helper 2 cells, or just be familiar that those are cytokines? Or do you just want us to know the ones on the board?
No, that's the ones. Those are the main ones I want you to. T helper 1 cells. Here we go. T helper 17. T helper 2. T reg cells. We're really. What we need to think about them with the T reg cells is they're going to associate, and so just think about this. The T reg cells are going to bind with the same antigen presenting cell as another T cell. And what's happening a lot of times is it's a self-reactive T cell that has escaped central tolerance, escaped the thymus, And that T-reg cell is right there to shut them down. And I really can't go into any more detail than that because I really don't get it. I'm not sure how it works, but it's very, very well known that these T-reg cells regulate inappropriate immune responses. Whereas, as I've said, antigen decreasing is a self-regulatory mechanism. Get rid of the antigen and the T cells stop working, the B cells stop working, and they will generate memory. That's actually one of the ways they think memory is generated is as the antigen levels decrease to such a low level, only the highest affinity ones will then be kicked into a memory state by some mechanism. I don't know. Dr. Latassi, you have questions?
I have a few words.
Oh my.
So I know this has been a lot, but I can tell you, sitting here listening to him, I'm thinking through the whole curriculum, and I can tell you it's going to come up over and over and over again. So it's very helpful. You need to get, if you have not had any immunology, get basic terminology down. Understand acquired and innate. Understand those. Those little videos I gave you should help keep putting this in perspective. So what we're going to do tomorrow, or let me tell you how some of these help. We can now measure the presence of HLA and HLLB in hypersensitivity reactions that will kill you. So especially in the Asian population, the Han, Chinese, have a couple of these present that if they take allopurinol, they get a hypersensitivity, they die. They take an anticonvulsant, they get a hypersensitivity reaction, it kills them. So now we can test for those. So that becomes really, it's very practical. Tomorrow we're going to talk about biologics. So we're going to talk about when the immune system goes awry, we're going to try and pinpoint especially these cytokines that are telling these cells to do something else and try to shut that immune off, that response off. So tomorrow we'll talk about IL-17, interleukin-6, TNF-alpha is a big one.
Yep.
Yes. So these biologics pinpoint one thing, and they try to change the immune system's response. Now, the problem with that is we haven't gotten really good because we don't know which ones are exactly critical. So if we shut off one, the immune system will kind of pop up and do something else. It's like if we use one drug to do something, we'll make it, you'll end up with psoriasis, or it'll become worse. So this is really good. It's the overall. I'm telling you, it will pay off in spades. But just work with wherever you know. If you know a lot, then add to what you already know. If you don't know anything, get your terminology down, and at least the big groupings down. And that's where we need to start.
And you'll tell her that some of those cytokine receptors, there's important molecules that are involved in signaling downstream. What are those that we might use as immunotherapy to try to shut down? You don't remember? It might sound like anus.
Oh, Janus.
Janus kinases. Remember?
We'll talk about those.
Yeah.
We have some drugs now that will hit those. We also can knock B cells out. Well, you can imagine if we do that, what other things we're going to do. So you can imagine if we knock out part of the immune system, then what becomes a problem? What can't you do? Can fight infection, and cancers become a real problem. So all of this will, I think, make sense as we'll just keep hammering it. It's just going to, these things will be said over and over and over again. Co-stimulation of T cells tomorrow, there's drugs that do that. So tomorrow we're just going to do broad introduction to biologics. I haven't finished up that lecture yet, so I'm going to plan to start at 9. If I think I can do it quicker, and you know me, I can do things really quick, right? No, I don't. We'll start at 10, so just watch for an email. Plan for 9, and if I think we can do it at 10, we'll do that.
So as far as concentration,